Hello everyone, welcome to my channel, Cardamom Mousery, formerly Brookie Cookie Creatures for the OGs out there. Today, I thought I would finally get around to making this Fancy Mouse Genetics introductory video. It's not going to be super fancy or college level. I specifically want to make this video for breeders and hobbyists and people who are just interested in mice out there who want to understand genetics from a breeding perspective, specifically for pets. There are a lot of videos out there for like feeder breeding or on just like the basic bare bones husbandry, but I feel like we are severely lacking in videos about like the genetics and how to understand genetics and use that knowledge of genetics when breeding to figure out what results you might get from a pairing, how you should pair mice together, or how you can select for better mice within those varieties over time. I will not be able to cover everything in this video because I don't want it to be 12 hours long. Because, I mean, once you get into it, Fancy Mouse Genetics, even from a hobbyist perspective, you can go as deep as you want. And for me personally, it's one of my favorite subjects in mouse breeding. So I tend to nerd out about it quite a bit. But for now, we're just going to go over, in this video, the basics of color genetics. And we'll go over the most common color genes in the Fancy, specifically, and how they work in a basic way, so that you can get a basic understanding. And then over here, oh, and I just made this cute little diagram right here to go over it, because I have made a couple other genetics videos in the past, but they're very messy and chaotic. And it's a lot easier to make mistakes when you're writing stuff out as you're going, so I still won't be editing this video very much. It's going to be a bit messy, a bit more like a conversation, so get cozy, maybe grab yourself a snack, a notebook if you want to sketch things out, and think about some of these ideas. But hopefully, throughout this discussion, this chill chat we have about mouse genetics, you're able to pick up on both the basics of genetics, and also get a grasp of the concepts enough that you can use them yourself. Because, as I say here, there are so many different colors, coats, and varieties within the species, which is very exciting, but it can also be pretty dang daunting. And understanding the basics of genetics will help you to be able to come up with pairings on your own, decide what mice you should breed, and it'll help you to know for yourself what you might be able to expect from a pairing, so that you don't feel the need to outsource that and look for answers elsewhere. And don't mind me, I am still recovering from a cold, but we'll get through this. One thing I notice as somebody who has been in the Fancy Mouse breeding space for a while, a lot of people who are just starting will be, I don't know, they'll come online and they'll ask, if I pair this mouse and this mouse together, what am I going to get? And the reality is that it's not that clear cut, and it's not something that, like, even if you pair two black mice together, you could get a Siamese or an albino, or a bunch of recessives, because recessives are hiding under the surface. So really, honestly, the only way to be able to predict and understand what pairings you should put together is understanding for yourself what you have, and the basics of what you may or may not inherit, so that you can come to some conclusions and do some test pairings to hopefully get the results you want. And like right here, I've got a whole bunch of cute little mice, just to show you some of the sheer variety that we have. Like there's a blue Angora, a black-eyed white dominant hairless, a color point beige, color point smoke splash Angora, blue point splash, blue point Texel, and then we've got a blue tan, a chocolate Angora, recessive yellow Angora, and an ivory. And those are all just a few varieties. Like, that's a very small selection of what's possible. Like, you've probably seen a dozen other varieties of mice. And by understanding genetics, if you have any of these two mice together, and you pair them, and you get your results, you'll be able to understand what you're getting. So hopefully by the end of this video, you will be able to understand at least the basic genetics and why these colors are what they are. First things first, though, you gotta understand the vocabulary. Sadly, it is important to understand the words that are used, and these words will be thrown around in the fancy, so if you have discussions with people in the mouse breeding space, or if you even watch my videos, you will definitely hear these words. So they're very handy to have an understanding of. Hopefully, I mean, I think most people have probably learned about them in high school, but just to give you an overview, in case you've forgotten, a gene or allele is the basic unit of heredity. All mice, and all animals, honestly, carry and pass on their genes to their offspring. Alleles are the different versions of a gene that a mouse might pass on. So that, for example, would be each of these individual things. So all of these little letters are individual alleles. A locus is a section of DNA that holds a group of genes with specific traits that control specific traits. A mouse will carry 2 genes or alleles on every locus. Just to give you like a visual example, this is not perfectly accurate, but you know, we all have chromosomes, various amounts for different species, and on each chromosome there are different sections, and those sections are what we call the locus, and each of these loci will have 2 alleles each from each parent. As an example, we've got the A-locus, and these are all possible alleles that might show up on the A-locus, and you'll have 2 of these. So you can basically pick any 2, put these together, and that can be an example of what a mouse might have on the A-locus. And then we've also got like the C-locus, and there are a whole bunch of different loci to keep track of. As you can see, we've got this nice list of the most common color loci there that we know about. The nice thing with mice especially is we have a lot of knowledge on the genes and their locations, especially in comparison to other animals, just because of how much they've been researched. So we're lucky in that way. Next up, we've got phenotype. Phenotype refers to the visual expression of a mouse's genes. So this is what a mouse looks like. It doesn't always tell you much about like what genes they carry. It tells you something, but it's always possible for a mouse to be hiding something. Like this mouse right here looks like a nice chocolate Texel, maybe. It's got a slightly curly coat, nice warm chocolatey color, you know, classic, predictable. So you might be like, okay, so it carries chocolate, and it carries whatever genes give you Angora, the long coat, and it carries whatever genes give you Rex. But that's not all. And that's where genotype comes in. Genotype is the genes a mouse carries, both the hidden and the expressed. So this includes the dominants and the recessives. And in this mouse, just to give you the basics without getting too crazy in details, we've got AA, which is, we know, recessive pairing because it's lowercase letters, and that gives us black self. BB, which is the chocolate right there. But then right here, we've got uppercase D, which is normal, with a lowercase d, which is blue. If this mouse had had 2 copies of the lowercase d, or blue dilution, it would be a dove, or blue and chocolate dilution mixed together. But it only carries it. So looking at this mouse, we wouldn't know that it carries blue, because it's hidden. So that's why the genotype is important, because if you paired this mouse right here with a blue mouse, you might get blues in the letter. You never know. So genotype is equally important to phenotype. Phenotype can be helpful, but it's definitely not the whole story, especially when you're breeding. Next up, we've got homozygous. This refers to when a mouse carries 2 copies of the same allele on a given locus. So like in a matching game, AA, lowercase aa, which is black self, we've got agouti, black tan. It's 2 alleles on the same locus that are the same. And heterozygous is when a mouse carries only 1 copy of the allele on a given locus. So as you can see here, they're mismatched. We've only got 1 agouti, 1 black self, 1 black tan, 1 agouti, 1 black tan. So they're mismatched on that one. And heterozygous and homozygous specifically refer to when you have, like, in this case, homozygous, the same gene on an allele, and the same gene, or 2 different genes on a locus here. So it's got to be on the same locus. Like, it wouldn't even make sense, but, like, if I had, like, A, and then I consider the fact that I had CH, the C locus and the A locus are not the same at all, so It's impossible. We wouldn't call this heterozygous because we only have one A and one C. Because that, I don't know. I feel like that's something that some people get confused about sometime. Just in case you were confused, this is about the locus. So just remember that they will be on the same locus, and it's whether or not you have matching alleles or not matching alleles on that locus. Hopefully that wasn't more confusing than not. But down here, we've got a little fun guy as an example, and we'll use him to help us understand these terms in connection. We've got dominant, which means alleles that only needed one copy to be expressed. These overpower recessives. And this guy right here is a brindle, which is represented by AVY. You only need one copy of the brindle allele for it to be expressed. And this little star symbol, you will see later, is a symbol that we use in the fancy and in breeding to basically represent an allele that we don't know about or that doesn't matter. And in this case, we know this guy's a brindle, but he could be carrying like normal black or extreme black or something else, and we wouldn't know because the brindle covers it, because brindle's dominant in this case. So we just put a little star to represent it might be something else, we're not sure. And then recessive are alleles that need two copies to express. And this is super simple. There are other types of inheritance, and a couple of these are technically a different kind of inheritance, but that very simple idea of dominant and recessive is the most important, so we'll just go over that now. I also usually get all of my other terms mixed up, even though I understand the inheritance of all of the different varieties. So, you know, we'll get through it together though. This little guy right here is a Siamese, and you need two copies of the recessive allele CH or Himalayan to get Siamese. So this right here would be our genetic code for Siamese. Fun fact about Siamese, it is actually a a temperature-controlled dilution. So the cooler parts of the animal are going to be darker, and the warmer parts are going to be lighter. So you'll have more pigment in the extremities, basically. Any part of the mouse will be cool. And that is the same for any other Siamese and other species. So Siamese cats, same story. I think that's really fancy. Siamese is one of my favorite varieties. I always have a few in my mousery for that reason. It's one of the oldest varieties that I've had. All right, now on to the fun stuff. Hopefully all of those definitions made sense to you. If they didn't, hopefully as we go along they stick a little bit better because I will be using the words. There's no better way to memorize something and get the ideas than to see it in action. So let's get started on the important genes. All right, so important genes, specifically the color genes. First things first, these are all of our different loci. We've got the A-locus, B, C, D, E, and to break the pattern, the P-locus. These are all the different alleles, and I've actually written them in order of dominance because, and this is where it gets a little funky, even though all of these uppercase A's are dominant and all these lowercase A's are recessive, there actually is an order of dominance, like a hierarchy within the dominance. And that's important to remember because let's say that you have two dominant alleles on the same mouse, like a Goody and dominant red. Knowing which one of these would come out on top will help you know what mouse you'll get if you were to breed an agouti and a dominant red together. And if you were to do so, the dominant red would win. AY is dominant red. And the agouti would be hidden. So that's important stuff to understand, and it will help you. So just so that you can start to get in your head, I've written them all in order so that you can get an idea. And that's the same for every locus. A-locus, right here. B-locus is the chocolate locus. C-locus is the dilutions, so these are all the C dilutes. They're very fun, we'll get to those. D-locus is the blue dilution locus. E-locus, also called the extension locus, is where we find recessive yellow, one of my favorite varieties personally. They're what my chaos line is based off of. And then we've got P for the pink eye dilution locus, which is a very weird one. Honestly, it's one of the funkiest ones, honestly, in my personal opinion. But by far the most important locus to understand And I'm going to say, like, if you can get down the A-locus, you'll have a million times, like, everything will be easier for you with breeding, knowing what you're going to get. Because the A-locus, as I say here, controls the distribution of yellow and black pigment on a mouse. So by understanding the A-locus, you will understand like how all of these other genes affect the mouse, and you'll have a better idea of what your end color is going to be. And that will make more sense here in just a second. First things first, helpful vocab, melanin. So the pigment that gives mice their color is melanin. It's melanistic pigment. This is true of pretty much every mammal, at least that I'm aware of. And it's also true for us. We have melanistic pigment. Melanin is not just black, though, believe it or not. There are different kinds of melanistic pigment. And in mice, they have eumelanin, which is black and chocolate pigment, both of those. And then we have pheomelanin, which is yellow and red pigment. And there are different dilutions in genes that give us various versions of these colors. But these phrases, these terms are honestly, like, they're just as important as any of the others. So I recommend trying to keep them in mind. It might take you a minute to get them straight, but pheomelanin, yellow and red, it might help you to remember pheomelanin, phosphorus. Honestly, that probably won't help, but phosphorus is a big part in what gives us the yellow and red pigment. And then eumelanin for black and chocolate. I'll use them interchangeably throughout this video. So if you hear me saying eumelanin, you can refer back and know I'm talking about black pigment. And if I say pheomelanin, I'm talking about yellow pigment. So, with that in mind, we know that when I say that the A-locus controls the distribution of yellow and black pigment, I mean that it controls where yellow and black pigment is located on a mouse. So these are all of our different A-locus alleles, and you'll see what I mean by the distribution here as we go. First up is dominant yellow, also called lethal yellow, because it is actually lethal when homozygous in utero. And what that means is any of the mice that are homozygous for dominant red will just be reabsorbed before they're even born. So you won't see them, and for that reason, you'll never have a homozygous dominant red mouse. They'll all only be heterozygous. And dominant red or dominant yellow is the gene that is most capable of giving you a rich, vibrant, warm red or yellow color. It's the easiest one to select for that really intense color. And it being most dominant, that makes sense. Because what you'll notice here is the more pheomelanin, the more yellow pigment a mouse has, the more dominant that allele is. So AY, dominant yellow, the most pheomelanistic of the A-locus alleles. Next up is AVY. This is viable yellow because they are not lethal in utero, so you can have homozygous brindles. And brindles, in comparison to dominant yellows, oftentimes have stripes. These stripes are agouti, and they can range anywhere from no stripes, which is an unmarked brindle, to an undermarked brindle, which has just a couple, to a normal brindle, which has like a nice stripe distribution, to an overmarked brindle, which has like a lot of agoutis, sometimes to the point where they look agouti. And you can also have like sooty brindle, where they have like, instead of stripes, they just have a line of soot along their spine. So in comparison to the dominant yellow, they've got a bit more eumelanin, and so they're a little bit less dominant. And then over here, we have our wild type, agouti. And I used to, when I first started breeding, I was like, oh, agouti, that's kind of boring. It's just wild, normal, meh. But now, bro, agoutis are some of my all-time favorites. I love them. I love them so much. A well-bred agouti is honestly amazing. They're so cool. And agouti mice, the fun thing about them is each individual hair will actually have two separate colors. It'll have both that eumelanin and the pheomelanin. So each hair will have both yellow and black, and we call that ticking, which is something that a lot of different species have in their wild coloration, because it's great for camouflage. So because each individual hair has those two colors, that distribution of yellow and black pigment, we get kind of the sandy look, and it's kind of like a half and half. So first we had basically no black pigment, next we had a little bit of black pigment, now we have half and half, black and yellow pigment, eumelanin and pheomelanin, and the distribution is each individual hair has the two split, so that's wild type. And next up is AT. And as you can see, the distribution of eumelanin and pheomelanin here is very fun. We've got the top half with eumelanin and the bottom half with pheomelanin. So top black, bottom yellow. So instead of the half and half that the agouti had, this one has half on top, half on bottom. And fun thing about black tans, or tans, is that fungally enough, even though this is a recessive, it's got that lowercase a, which is how we represent our recessives. Even though it's got that, and here I'm going to change this brush so I can just, yeah, that's better. Even though it's that lowercase a, the yellow belly will actually act more like it's dominant than recessive, but only the yellow belly. The black top still acts like a recessive. It almost acts just like black self. But for that reason, if you have black tan AT and agouti, which is dominant, the belly of the agouti will actually become tan. So you'll have a yellow belly with an agouti top if you have black tan and agouti, which is kind of funky because yellow man, it's just so dominant. It's powerful. So that's a fun little specific thing to keep in mind if you breed tans, is if you breed a tan, even if the result is heterozygous for tan and you've got another gene present, the tan will still show up. Now, you don't really see it on dominant yellow or brindle because they're yellow and the belly of a tan is yellow, but technically it will still be there. So that's a very fun thing. Distribution, once again, yellow pigment on dominant yellow everywhere. These guys, a little bit of eumelanin mixed in. Agouti, half and half. Tan, literally half and half. And next up is black, non-agouti. And these guys are all black except for a little bit of tan pigment or yellow pigment in their ears, around their vent sometimes around their muzzle. And we call these fault hairs in the fancy just because if you're breeding for show quality mice, your goal is to get a mouse that's as dark as possible. And those tan fault hairs, of course, are not black. But black self will still allow a little bit of that pheomelanin, that yellow pigment through. So you will end up with a black self mouse with just a teensy bit of that pheomelanin lingering. But next up is extreme black. And it has no pheomelanin. Now, extreme black is actually one of my personal favorite genes, my favorite A-locus alleles. I think extreme black and agouti are my two top favorites in this locus. But a common misconception with extreme black, and you'll see this as you're researching, is that it turns yellow fault hairs to white or clear. It does not. It does actually fully dilute yellow pigment to black pigment. It completely removes pheomelanin. So all of these other ones, you have a lot of pheomelanin, slightly less, half and half, a tiny bit of pheomelanin left. Extreme black has none. So this will be a fully eumelanistic mouse, fully black. But here's the kicker. These guys, in spite of having no yellow pigment, can still have poor quality depth of pigment. So you can still have like a pale, sad looking extreme black. And just to give you like a demonstration, you can probably see there's a difference between these two colors, but a black self can look like this color and it can look like this color. And the thing that's going to give you the really nice dark black is selection. So you still have to put in the selective work to get a dark pigment on an extreme black, but the benefit of extreme black if you're going for show quality blacks is you don't have to breed away from the fault hairs that you would have to breed away from with just a normal black. So that is the A-locus. As you can see, it goes in order from the most pheomelanistic to the least, and that's the order of dominance in these guys. And as we go on and you see the other loci, you will see just how important it is, because all of these other loci, they act on the A-locus. So they act on that basic foundation that the A-locus set up for us, and they all dilute these two pigments in different ways. So like if you have this mouse, the yellow pigment and the black pigment are both going to be affected differently by the B-locus, the D-locus, and on and on. So without further ado, let's move on to the B-locus, which is a lovely locus that controls chocolate.